Hello, Alysson Muotri here. I'm a professor at UC San Diego in pediatrics, cellular molecular medicine. I'm also the director of the stem cell program here at UCSD. And this video is about the NASA town hall for animal research, thinking like 10 years ahead. And one idea would be to use chimeric animals. These would be animals with human cells transplanted in a chimeric way. Could be individual human cells, or could be organoids or different brain regions that are transplanted inside the animal. And then you use the animal to send to the International Space Station to understand what's the impact of the space environment, microgravity, radiation. So what are the advantages of that? I mean, currently animal models, they have the advantage of it's the entire body, it's very physiological, but they are very far from humans. I mean, especially mouse, small rodents, they are apart millions of years of evolution, so our brains are totally wired in a different way. And even our closest relative, the chimpanzees, we have different in size, different in wiring of the brain. So the translatability of the animal research to humans is a problem. So what is the alternative? Well, the alternative would be, well, let's send human cells, human brain organoids. That's what we are doing right now. We are working on systems where you can study the impact of spatial environment directly on human cells and even human brain organoids that somehow recapitulate, at least partially, some of the aspects of the human brain. Biggest advantage, you are working with the human cells, this is the human material. Disadvantage, well, it's outside the body. I mean, these are cells in a dish or floating in a 3D structure that doesn't have, for example, a skull if you're working with brain organoids. So the chimeric approach would allow you to combine both fields, so you can study the impact of the environment in human cells inside a live incubator, that is the animal, and then you can use small animals such as mouse to do your transplantation. It also allows you to do different types of chimerisms. You could transplant cells in different parts of the animal and focus on comparing the impact on human cells as well as the neighbor mouse cells. So you take in consideration all the differences in metabolism, things like that. So we will gain a lot of information by doing that. First, you don't need to engineer an incubator to hold the human cells as we do today. There's lots of engineer involved on keeping brain organoids or human cells in space, surviving in space. So you're just using the mouse as an incubator. And you're also going to gain more on the physiology, because it's most likely those cells or for example, neurons will be fully integrated into the brain, so you can study connectivity, what's the impact of that specific cell type on the anatomy, physiology of the animal. So those would be definitely something that we're going to gain that currently we don't have. So in a nutshell, I think that's the idea, developing chimeric animals so we can take advantage of their bodies and the human cells in a single experiment. That's it. Thank you.